Thanks, everyone. Welcome to the April 2026 webinar of the UK Freshwater Quality Research Programme. So my name's Joe Holden and I'm one of the programme champion team along with Pippa and colleagues here at Water at Leeds. There are 6 projects now on the programme. There's been a recent addition and that recent addition is focused on Loch Ness in Northern Ireland. And then we have 5 other projects which have been running for three and a half years roughly now, or just over 3 years. So EcoMix, Pacific, and then LTLSFE, which is a large-scale modelling project, MOT for Rivers, and finally Quantum. And what we do in these webinars is we have updates from each of these projects at sort of regular intervals, and then we have a bunch of other presentations in our webinar series as well, from sometimes an international presenter, sometimes another project, or something else pertinent from the policy and practice community. So it's a really great chance. So please do look back at some of the previous webinars and keep updated with future ones. And the benefit, of course, of joining our webinar live is that you can ask questions and engage with the presenters in dialogue. So we record the main presentation itself, but then later, after the presentation's ended, we have an unrecorded Q&A session where you can, at your kind of discretion, raise issues and questions which are not then recorded. So looking forward, we've got a couple of webinars in June and then one in July before the kind of summer, little bit of a summer break. So do put those in your diaries and do join live if you can, so that you can engage with the presentation as much as possible. And of course, as usual, if you want more information about the programme or want to contact us, there's a number of ways you can do so. There's an email address, which is just freshwater@leeds.ac.uk. There's obviously a programme web page, and there's some other kind of QR codes there that take you to our web page, and also take you to some biosecurity training as well, which is very, very important when we're sampling freshwaters to ensure that we're all being responsible. So today, fantastically, we've got Professor Penny Johns from the University of Bristol, who's going to give us some updates on the Quantum project, and I think she's going to provide a bit of a deep dive into some of the components of the project, which have got some exciting and important results. So very happy to hand over to you now, Penny. Off you go.
Thank you. Just share the screen. Oh. Can you just confirm that you can actually see that? Joe,
Yes.
Yeah, is that okay? Brilliant. So I'm going to give you an overview of where we are with the Quantum project, which is quantifying the nutrient enrichment, pathogenic, ecotoxicological and organic pollution, actually, impacts of livestock farming on UK rivers. So the programme is structured in this way, and usually we end up with these webinars talking about work packages, but we're starting to move away from talking about individual packages of work and starting to think about how we're actually tackling and how we're answering the main themes we set out to address. So there are 3 themes for the Quantum programme. The first is exploring how the environment basically affects the sources and processes by which pollutants from livestock farming enter, mix and are transported to river systems. Then we have theme 2, which is looking at the impacts of those contaminants on river ecosystems in the UK, both from ecotoxicological and a nutrient enrichment perspective. And finally, theme 3 brings it all together and brings the evidence into a space where we can start to develop better plans for adaptation, mitigation or detection of these risks as they drive UK river water quality. And today I'm really going to give a deep dive in particular on theme 1, because this is the one where we've got most advanced with the analysis of the data that we've collected. So I'm going to show those to you. I'm going to tell you the conclusions we're starting to draw from those data sets, and then I'll give you a quick window, if we have time, into theme 2 and theme 3 and where we are with those parts of the programme. So theme 1A is looking at these risks from livestock excretors. So livestock excreta include dung, urine, farmyard manure and slurries. If you wonder what the difference between dung and farmyard manure is, there's the dung is fresh, voided that day in the field or in the yard. And we collected these from sheep, dairy and beef cattle farms at 3 different spatial and temporal frequencies. So the first of these was the seasonal variation in composition. So we had 6 main research farm platforms and 1 farm, Eberlands, which is very close to the site where we did our subsequent experiments. And in each one of these, we collected all 4 forms of excreta from these 6 platforms across England and Wales, both in the winter and the summer. So we could look at seasonal variation in composition. You know, does the risk vary by season? Then we also and our second part of theme 1, looked at the monthly variation in the composition of slurry from 1 farm at our Rickford site in the Congressbury catchment. And then finally, we then did a national sampling programme where we visited 60 farms once, headwater streams only, so no sewage sources in those catchments. And we've looked at just the dung alongside water and sediment quality. So all of the samples were analysed to determine their nutrient, ecotoxin, pathogen and organic matter composition. And what we're using these data to do is to answer the question of how risk to UK rivers varies in space and time between farms, between livestock types and production systems, between excreta types and also between contaminants. So can we work out how that risk shifts in space and time? So just to outline a little bit more about the national sampling programme. So here on the left, we've got a graph taken from the Animal and Plant Health Agency. It's this population density for the dairy and beef cattle in July 2024. And you can see that they're all over primarily on the left-hand side of the UK. So this is a fairly high population densities in Devon and Cornwall, in southwest Wales, up in Anglesey, through the west of England, into the Lancashire, Cumbria area, and up into the southwest of Scotland in particular, and the middle section of Scotland. But if we look at the central map, you can see that this is where the sheep and goats are, primarily sheep in this population data. You can see that the population density is much higher. We're looking at 10 times higher than it is for the dairy and beef cattle primarily, and those are filling in the gaps in the west. So if you haven't got dairy and beef cattle, then you've got sheep. So anywhere we've got upland environments, you can see the Rib of Wales, you can see Exmoor, Dartmoor, you can see the Pennines and so on going up into Scotland, right up towards Aberdeen. So this is where they're distributed, and therefore our sampling sites for the Quantum National Sampling Programme, NSP, which we did the following year in June to August 2025, follow those patterns in order to capture those patterns. So we had 60 sites, 20 sheep, 20 beef and 20 dairy cattle. It was quite difficult to find a catchment that was only beef cattle because often you had beef and sheep alongside each other on the same farm. So we had to be very careful with how we selected sites. Most of the site visits were unannounced. So we arrived, we sampled and we left because we wanted to see farming as it is and not how it might be presented if there was an inspection going on, for example. At each site, we collected samples of water, stream sediment and dung. We haven't got the funding at the moment to analyse the stream sediment samples, which is a shame, but we've got those in cold storage in case we come across a pot of gold in future. We have completed all the nutrient analyses and processed the data for those samples already, and the pathogen, ecotoxin and organic geochemistry analyses are now complete, but we're still processing the data, so we can't show you those trends yet. If you're wondering about the geographic representation, we had a total of 16 sites in Scotland, 21 in Wales, mostly because it was easier to get to than it was to get to Scotland. We had 9 in the north of England, 3 in the west of England. We tried for more sites there, but many times we arrived and we found the streams were running bone dry and there was no water left to sample, and then 11 down in the southwest of England. We also collected samples from a few sites in some of the other freshwater programme catchments, so we can share the knowledge from this project with the rest of the freshwater programme. So let me tell you about some of the trends that we've seen, and I'm just going to give you examples. I can't possibly present all of those data in a short presentation like this. So first of all, nutrient composition in urine, and we've discovered that this varies both by season and livestock type. So at the top of this this slide, we can see we have the data for nitrogen. We have total nitrogen, so that's all of the nitrogen in the sample, including nitrate and ammonium and the things you may be more familiar with, and then also urea, which we've discovered is highly bioavailable to freshwater biota. So we've looked at that one in particular, and the key conclusions we can draw is that in winter, the urea and total N concentrations are highest in sheep urine. The total N and urea concentrations are almost equally high for sheep and dairy cattle. And in the summer, total nitrogen concentrations are highest in the beef cattle urine. So this is in the raw material, not in the stream. And then if we look at the phosphorus data, we've got phosphate, which is the thing you may be more familiar with, but also total phosphorus, where in most systems, total phosphorus is about 70% higher than the first phosphate. So the total phosphorus and phosphate concentrations are again highest in the beef cattle urine in the summer. So that mirrors what we see in the data for the nitrogen. The total phosphorus concentrations are also higher in dairy cattle urine in the summer. And then total phosphorus and phosphate concentrations are highest in sheep urine in the winter. So we're seeing these variable seasonal effects. It varies by livestock type as well. Now we can also look at some of the more detailed data. So here we just have a selection of determinants. DIN is the total oxidized nitrogen, so that's nitrate plus nitrite, and ammoniacal nitrogen together, so that's the green bars. In the middle we've got the orthophosphate concentrations, and on the right again we've got the urea. And we can draw a number of conclusions by digging into the data, because you can see they vary both by the livestock and excreta type. And what we found is that they present risks of nutrient pollution, which varies by season, by livestock and excreta type. And so if we want to manage those risks, we need to be thinking about when we're sampling this material, when we're sampling streams, and how we're actually going to gather the correct evidence. Moving on to that monthly sampling data set. So these were the slurry samples we applied to our field experiments, which I'll tell you about in a minute. And we've been able to compare those. So the nitrogen in our slurry monthly data is shown in pink on the left. The phosphorus P2O5 slurry monthly data is shown on the right in blue. And you can see that those data sit firmly within a much broader range of farms that have been sampled across the UK, where the samples have been sent to NRM Kwood under RB209 for analysis. And so you can see we have representative samples, and what we've concluded from this is that they fall within the expected range reported by NRM Kwood. So let's talk a little bit about profiling the other things, not the nutrients, which you might be more familiar with. So we're going to talk about chemicals of concern, or COCs, which are also referred to as ecotoxins. So these have a toxic effect on the biota. There's antimicrobial resistance and the genes associated with transfer of that material in microbial communities. And then there are pathogens like E. coli. So an initial screening technique developed by the University of Bath partners in this project screened for over 198 target compounds. We've got antibiotics, we've got other pharmaceuticals and biomarkers, we've got the endocrine disruptors and the hormones, and then we've got fungicides and antiparasitics, all of which are routinely used in livestock farming. And the ones underlined in red are the ones that we actually detected in that set of just six farms that we've sampled so far. So we're still looking at these data for the broader national picture, but this is what we found in those 6 farms. And as you can see, it's quite a lot of samples, particularly the endocrine disruptors and hormones are very commonly found in these things, and things that are unusual. So you might think when you go to the doctor, trimethoprim is a drug you might get if you've got tonsillitis. You also find it in animal excreta. Things like caffeine and ibuprofen, you might think they're only associated with humans, but in fact they're also associated with livestock farming. So just taking one set of samples then, so here are 3 farms from that selection of 6. One has got beef and dairy, one has got just dairy, and then farm 4 is also just dairy. And this is the concentrations of all of the different compounds in those individual samples. And you can see there's some commonality. For example, if you look at the yellow bar, you can see that appears at a similar height on all 3 of the farms, but some of them are much more specific to the farms. So things like tiamulin and lincomycin, which you can see are in pink and in a sort of a teal colour on those graphs, are actually only found on farm 2. So some of the compounds are found specific to a farm, some are found everywhere. And we're working our way through that data set. We can also look, for example, at a single set of samples for winter urine. So this is the winter samples from those 6 farms, and these are the concentrations, and you can see how it varies between beef and dairy and sheep. So sheep have relatively few of these compounds present, just the clarithromycin in farm 2 in the winter in this sampling programme. But other farms, for example, in beef farms and also in the dairy, we're seeing these pink and teal coloured bars popping up. We are currently working on a method which was originally developed for sewage effluent, and we'd probably need to do some analytical method refinement in future projects to reduce the limits of detection so we can detect a wider range of compounds. We know they're there, but we can't quantify them because the methods are not quite sensitive enough for some of the compounds. And just to show you that this is a plot of all of those data, just for the 6 farms. And I put this up really not to ask you to look at it in any detail, but just to show you the level of resolution of information that we've collected across this programme. And we are digging into all of these data sets in order to try and tease out those broader scale patterns which will help to inform policy. We also did a lot of work on the pathogens, and the one that stuck out most as a key finding was E. coli. We found this in every single excreta sample that we've analysed. So this is a log of the genocopies per gram of material analysed, and you can see it varies there across those systems. And the E. coli detection is highest in winter and in dairy dung and farmyard manure in particular, but it's also found in the beef and in the sheep as well. And as my colleague Davy Jones, who's part of the team, said, he's at Bangor University, and he said you can imagine that the livestock effectively are E. coli incubators on 4 legs. So wherever they are, you would expect to see this present as a risk to freshwaters. We also have AMR, so antimicrobial resistance risks from livestock excreta. So here, just showing one image, again, just for the winter samples, of the abundance of different AMR genes in those samples. And you can see in particular that the tetracycline and then the aminoglycoside in particular are highly represented relative to the other risks in those samples. Okay, so we can also quantify this risk to freshwaters. So here's a worked example for one of our slurry samples. So it's actually from 3 different farms. Here are 4 different antibiotics that we've looked at, and you can actually calculate the risk quotient where anything over 1 implies there's a high risk of transfer and AMR development in those systems. So we found 68% of the chemicals of concern in slurry samples exceeded the thresholds for freshwaters for AMR, with a high potential to select for AMR in freshwater microorganisms if the slurry is overspread, so it sprays over into a stream or close to stream margins. So in terms of the AMR work, we sent the samples off to be mapped. We used qPCR to screen for 96 genes, including both AMR and what are known as mobile genetic elements, MGE. These are the things that allow that antimicrobial resistance to be transferred from one microbe to another. We looked at the relative abundance, as you've seen in the previous slide, where the aminoglycosides and tetracyclines were highest in all of our samples. We found many clinically relevant genes in the samples, and amongst the MGEs, the profiles did vary between sample types and were generally higher in sheep and beef compared to dairy. So what about the national scale information on risk? So these are early results just from the nutrient chemistry and the pathogen analyses. So we know, just as a quick, a note, the E. coli was found in all of these farms in all of the samples that we collected. So what were we looking at? So beef cattle, and we might think that when we look at a beef cattle farm, it's very idyllic. We see them lined up on the banks of the rivers, they're often in the rivers, having a lovely time. But the reality is that in the winter, it looks much more like this. And actually, these ones on the bottom are all photographs taken from some of those 60 sites visited in the National Sampling Programme. And you can see the turbidity, the direct voiding of dung in the stream that is caused by this unlimited access to surface freshwaters. So here are the data for the nitrogen, phosphorus, dissolved organic carbon, that's DOC on the bottom right, and urea concentrations. And I've ranked the data here from farm 1 through to farm 20, and then we start with them ranked by nitrogen species concentration. So the top of the bar is the total nitrogen concentration, and then you can see within each bar how that's broken down into the different fractions. So for some of the farms, for example, on the left of this graph, in the top left, you can see that the majority of that loading is present as nitrate. But if we move to farm 4, then you can see here that actually there's virtually no nitrate in there, and in that system it's still got a very high nitrogen concentration, and below it you can see the urea is extremely high in this system, and the rest of that load is made up of dissolved organic nitrogen, particulate organic nitrogen, and then ammonium, and all of these are highly bioavailable to the freshwater biota. So you can see the data spanning out then in terms of the different phosphorus fractions as well. So we certainly have a real problem with site 1 in terms of these concentrations. And this is data for the streams, not just for the excreta. Okay, so if we then look at this, you can see the distribution of the localities of those sites. So if it's a West of England site, we only managed to get 2 of them into this data set, then they are the top 2 sites there, and we do have a real problem with the amount of water available to dilute livestock excreta and other contaminants in these streams where so much has been abstracted for dairy production in the region. If you look at the red ones, those are for the southwest of England. The purple ones are for Wales. The green ones are north of England, and then the black ones are Scotland. And what you'll see is that that risk is variable, but it's distributed equally variably across the UK. We can't look at these data and say, oh, Scotland's better than or worse than Wales, or vice versa. In fact, they all have bad and good sites in terms of the risks to freshwaters and what we've actually observed there. So let's think about dairy farming practices. So again, it's usually very bucolic. This lovely lady in the middle photograph there is wearing one of those lovely robotic collars. So, you know, when she wants to go and be milked, her robotic collar allows her to go to the milking pile whenever she wants to. So that all sounds lovely and good for animal welfare. But the reality is that it's often much more like this. And again, these are photographs taken from the sites visited unannounced during the summer months. And if we think about the other type of dairy farming where we're indoors 24/7, we have overfull slurry lagoons. That's the one in the second photograph along there. We have these fields which are heavily fertilised and heavily amended with slurry in order to produce enough grass to take off, which has been happened here. It's why it's bright green, in order to produce the food for the animals which are inside 24/7. We also have legislation in the UK, and in England certainly, farm rules for water, where we're not supposed to spread slurry within 10 metres of a watercourse. And I don't think anyone's really stopped and thought about what does 10 metres from a watercourse actually look like. This photograph is from our field experimental site, and you can see just how close that tractor is to the stream, and they're using best available technology to do precision application of slurries. This is not somebody broadcasting it around, as you might see more commonly in images like this one, which is again from another one of the sites that we visited. So if you look at the dairy data, you can see that the nitrogen is even higher. It's actually double the concentrations that we saw in the streams where we have beef cattle farming. And again, you can see that variability of the composition of the material, and also of the risk as it varies geographically across the UK. And similarly, we have several flagpoles for urea on the dairy farming sites, which is a real concern. Let's finally think about sheep. And again, you know, cute and cuddly, they often look like this. They're marvellous, they're very photogenic. But actually stocking densities can be really high, particularly where they've been brought into lowland environments. And actually in the winter months, we have things like this for poaching around the feeders. We have them directly accessing streams. That's commonplace across all of the systems that we looked at. Again, these photographs are from sites that we actually visited. And one of the big problems of sheep farming, and if you look closely on your screen, hopefully you can see all the white dots there, all over those bright green fields. There's a maze of drainage networks running through there where the sheep either have direct access or can get within 1 metre of a watercourse, and so therefore this material is very easily flushed from land to stream. So concentrations in these sheep farming streams are lower than they were for the beef and the dairy cattle, although site 1 for urea is again a real concern. It's extremely high and would be toxic, as well as generating a nutrient enrichment effect there. And you can see the distribution of those sites there across the UK. So if we start to think about what that means, and I would like to say right from the outset, These are either the correct environmental quality standards, the footnotes there you can look at a later stage and work out which bits of legislation they come from, and some of them are calculated from other thresholds which are in place, but they don't yet exist in the UK legislation. So just treat these with caution. But basically the message is that you've got the mean plus or minus the standard deviation, comma, and then the maximum concentration observed. And if you look at those data, if it's yellow, it's in excess of the threshold. And I would just like to draw your attention to the dairy cattle site for ammonium. So that's the second row down and the second column of data in. And if you look at the maximum there, the maximum concentration is 72.8 milligrams per litre of ammonium N. I didn't believe the number. I made my technician rerun the sample 3 times, but it turned out that it was probably ammonium chlorate that had been used to clean the indoor dairy parlour there, and effectively the whole stream was contaminated at an extremely toxic level. So we've removed that from some of the data sets we're working with because it's an abnormality. Okay, so key findings so far from the national sampling, and I have to say this did come as quite a shock. So no sites had a 10-metre buffer zone for slurry applications. Only one of the 60 sites even had a 5-metre buffer zone, which was a Scottish sheep site that had been in receipt of environmental stewardship funding, and it was an SRUC farm. So we believe that mitigation through buffer zone use is poor. It's universally poor in UK headwater catchments. It's not being used. Beef cattle farming was visibly the dirtiest. It had the highest particulate nitrogen and phosphorus loading and the highest phosphorus loading overall of all livestock categories. Direct stock access to streams was commonplace for beef cattle. 100% of sites they could get directly into the stream, and they did. And for sheep, about 90% of sites, the sheep had direct access. 9% had 1 metre fencing, usually on one side of the stream only, so they could get in from the other side, and only one had even a 5-metre buffer. So mitigation through fencing is also poor in UK headwater catchments when you move away from those sites where perhaps you've got a local interest group, a rivers trust or whatever, working with local farmers to put more in place. 75% of dairy sites had no outdoor grazing and high stocking densities at some of them. Most had direct access to the streams or a 1 metre fenced out of watercourses. Direct access to streams zeroes the options for mitigation of contamination fluxes to streams. So if we can't keep the animals out, we might as well forget about any other mitigation in a catchment because they're going to avoid that material directly, neat and untreated into the watercourse. Outdoor dairy sites often had really low flows, reducing dilution capacity. Indoor dairy, when I went looking for sites in the west of England, probably went to about 15 sites, ended up only being able to sample 3 of them because the rest were dry to at least 3 tributaries downstream from the source. And that came as quite a shock. So maybe this is kind of one of those hidden problems that we haven't really thought about in terms of the way in which we're producing dairy products. The indoor dairy systems also had really high nutrient concentrations. I've mentioned this one already, and we're awaiting the ecotoxin, organic geochemistry and pathogen results with interest to see if they follow the same trends as the nutrients. Loss of dilution capacity is a hidden cost. Dairy farming had the highest nitrogen concentrations, typically from the 24/7 indoor systems and also from the high intensity outdoor grazing farms. Sheep production is typically less intensive, but in sensitive upland catchments, moorland and mountain streams, it can present a significant organic enrichment of the streams. So policy advice and mitigation strategies that we're going to be testing in our modelling are likely to focus on these discriminations between the livestock types, but also the livestock production and management system that we have in place and the way in which stock access water to farms. So key conclusions. Livestock excreta are a key source of organic and inorganic nutrients, with risks varying between these different variables. E. coli was present everywhere, and livestock excreta therefore presents a significant risk to both ecosystem and human health if it's washed into or directly voided into freshwaters. So that's the first part of our programme. I hope that wasn't too fast. I'm just going to keep an eye on the time here. So I'm just going to tell you a little bit about thinking about how it moves from land to stream. And we're exploring the role of buffer zones. So we have 4 questions relating to how that material moves from the land to stream and how land management and environment modify that. We've set up an experimental site in the Rickford Brook, Somerset experimental catchment, and there's the map there showing the various features of the site. I'm going to be focusing on sites 5 through to 8 in the subsequent presentation. And we set up this experiment in that field between numbers 5 and 8 here, looking at how effective buffer strips are in reducing these contaminant fluxes from land to stream. So in strip 1, we have that buffer strip 40 metres away from the stream. So does it help if we make it wider? And in buffer strip 2, it's 10 metres from the stream. And then between them, we have control strips where we have no slurry being applied to them. So this is how we applied the slurry. We were very careful to do it the best way possible, and we did it at the same rate as we would expect to see in an NVZ. Now the site isn't in an NVZ, it's close to one, but we thought we'll try and do it the best way possible under the Farming Rules for Water and the Nitrates Directive legislation, and see whether we can actually get these buffer zones to work. So conclusions. Livestock excreta was going to be a key source of organic enrichment at Rickford based on that slurry analysis. The E. coli were present in all the slurry samples. We found macrolide antibiotics and phytoestrogens, so things that are going to generate estrogen mimicking impacts on the freshwater biota in all of the samples, suggesting the opportunities for AMR and endocrine disruption. Chemicals of concern profiles represented similar practices that we'd seen in our theme 1 slurry samples, and the slurry amendments we thought were going to be due to a key source of nutrient enrichment, pathogen, ecotoxin and AMR in the Rickford stream, particularly if they're washed in during wet weather. So we set the experimental up. We applied the slurry wherever you see a red star. We have a daily rainfall record, not for the site, but for the top of the Mendips. So some of the events you see on the top of the Mendips would not have appeared lower down at the site where we were working, but this is the best available record. And then the green boxes show when we actually try to do our monitoring. So we monitored in experiment 1. You can see if you look at the site flow, there's nothing going on the flow. So those are base flow conditions. If we look at site, the second one, again, we somehow managed to stumble into the only period when it wasn't actually flushing in experiment 2. So we've got base flow conditions sampled then as well. And then the third one, experiment 3, here we managed to capture some storm flow responses in relation to the slurry amendments. And what was surprising of the evidence that's come out of that, and again, we're really working on this at the moment, so I'm just showing you one example of the data. Here is the nitrogen chemistry response to the slurry amendments. So 10, 11 and 12 are the 10th, 11th and 12th slurry amendments applied to the plots, and the yellow bars show the peaks in the stream concentrations of nitrogen in relation to rainfall events after slurry has been applied at site 7, which is the 40-metre buffer strip, and the green peaks are the ones at site 8. Sites 6A and 6B are upstream from the slurry amendments, and we don't see those same peaks. And what that tells us is that the buffer strips, even if they are 40 metres wide, are not able to trap these incidental losses to the streams. So buffer strips are not working. So key conclusions, we found no differences or impacts of either the 40 or 10-metre buffer zone width on stream nutrient concentrations. In experiments 1 and 2, under base flow conditions, we saw big incidental losses getting to the stream under experiment 3, and there was no evidence to suggest that the buffer zones were able to trap these fluxes from livestock farming to streams. We're still processing the pathogens data from the field and soil column experiments, but we did find it was present in all sites and in all experiments, and that buffer zones do not reduce that flux. And some of our work on soil columns has suggested that the pathogens actually move largely unhindered through soil systems. They don't get trapped the way some chemistries might get stuck to the soil. They move through very efficiently. So once they're on the soil, they are likely to eventually flush to streams. Finally, the molecular scale analyses are now complete, but we're in the middle of data processing, so we can't tell you anything about whether that's also true for the ecotoxins, chemicals of concern and AMR. So I'm just going to give you a quick update on the, I don't know where that's there, it'll get wrapped up in a minute, on the work we've done to look at the impacts of these fluxes on the biota. So the ecotoxicology work started later. I'm sorry that is in the way. I don't know how to get rid of that. So we have in situ duplicate caged exposure experiments at Rickford. we've put in these chemical passive samplers, the photograph on the bottom right, and we're running a whole series of biological measures which have allowed us to examine the impacts on two biotic groups, on stickleback, three-spine stickleback, and then the freshwater shrimp, Gammarus. The stickleback were maintained in relatively condition throughout condition throughout the experiment. The Gammarus had a really poor survival rate of only 37%, so we've collected some wild Gammarus, and that work is still ongoing. The second one is looking at this material as a source of nutrient enrichment, and so we've used stable isodosing to investigate how this material is taken up by the stream ecosystem. So we have these jars submerged sort of shoulder deep in the stream. We have 3 for each chemical treatment. We have 6 different organic chemicals, ammonium sulfate, nitrate as well, potassium nitrate, and then we have added to the jars the stream water and isotopically labelled compounds for all of the organic material we're interested in. We've also got slides coated in biofilms, and then we've got bunches of bryophytes, which are all native to the stream, so they're cultivated from the stream. And then we run that over a 4-day period, and we collect the samples. We also did an experiment adding snails at the end, and we demonstrated that 70% of what we added as an isotope that was taken up by the biota, the primary producers, was consumed by the heterotrophs. So it's gas whole food web scale interactions. And here are just some of the data. So here are the data for biofilms. You've got the ammonium on the left, you've got the urea and the glucosamine, glycine, glutamate amino acid mix. So glucosamine is an amino sugar, glycine, glutamate, and then the amino acid mix are all amino acids. The urea and the glucosamine appear to be behaving in the same way, where the blue line, which is the nitrogen uptake, is in blue, and then the carbon is in red. And we can see that, for example, for the urea, even after 4 days, the uptake rate into the biota is still going up. The same is also true for the glucosamine and also for the glycine. So these are highly desired, rapidly consumed compounds. And you think, well, maybe that's true for biofilms because, we expect microbes to work quickly. But actually, if we look at the data for the bryophytes, we find we get exactly the same patterns. And if anything, we get even higher rates of uptake of the amino acids, particularly the glycine, glutamate, and the amino acid mix in these systems. So what we're demonstrating there is the importance of this material to the stream biota. It's used at least at the same speed or in excess of the speeds of the inorganic compound uptake. So here are our key conclusions from that. LDOM, livestock-derived organic matter, is taken up by both the biotic groups at rates equal to or higher than those for inorganic N. Nitrate is actually the least favoured as an N resource. The uptake rates for nitrate, which I haven't shown you there, are up to 10 times lower than for the other compounds. Within 4 days of addition, 70% of the labelled carbon and nitrogen moved into the snail biomass. So these additions from livestock farming are likely to drive responses in complex freshwater food webs if we allow them to transfer into streams. We can also see marked differences in the way in which they're using different compounds. Climate warming had variable effects, so we did a climate warming experiment using this approach. Some compounds were taken up more quickly, but others showed little or no effect of warming. And we did see a much bigger buildup of phytoplankton biomass in the jars, which means that they're consuming some of the isotope that we were actually adding. And we're using the results of this and the work from the ecotoxicology experiments to inform scenario design for our national scale modelling. So I'm going to quickly whip through just a few slides on the modelling, just to show you what we're doing. I won't spend a lot of time, but if anyone has got any questions, the good news is that we have a paper out in the last couple of weeks, which will give you time to read about this at your leisure. So we have 3 tiers of modelling. We took one of the models, which is called field mouse, and this computes the runoff, pollution transport and contaminant losses across land surfaces through river networks. And what it does, it produces 3 layers of information in a GIS-based model. It tells you the travel time, it tells you the importance of the source, and it tells you the subsequent pollution risk to freshwaters. And we set it up and tested it in 3 UK catchments. So we have Congressbury Yeo in Somerset, which includes our experimental site, and then 2 more that are located in Cumbria. And so I'm going to show you just some examples from Congress Brio to make sure we've got time for questions. So this is an example of the type of outputs it produces. So on the left, this shows the potential 10-metre buffer zones that we might be able to put in place practically. So they haven't got housing in the way, they haven't got a railway cutting in the way. These are places where we could, in theory, add buffer zones to this catchment. And on the right, you can see that simulation of the risk of a contaminant flux from land to stream. And by layering all this information together, we can show that the darkest red bits are the ones which have the highest risk of transferring contaminants from land to stream. We ran the model. We haven't yet put our own data in from the field site, so this is going to look a lot worse once we do this, but using reported data from the literature, we put these 10-metre buffer zone strips in, and if we did ultra-low nutrient applications to all of the land upstream from the buffer zone, we could get a maximum of 9.3% reduction in contaminant flux from land to stream. But where we applied the slurry at the rate comparable to that applied in the work package 2, which is taken from the NVZ rules, immediately upstream from the buffer zones, the model suggests that we would achieve only a 0 to 1% reduction in nutrient loading to the stream for a 10-metre buffer zone, and maybe 1.5% reduction if we increase the buffer zone width to 40 metres. So it's not looking good for buffer zones. Finally, getting to the national scale, we have this type of capacity in the models to run scenarios. So we implement a land use change strategy, say taking medium soils with a short travel time to the stream and reverting them all to grass. So putting them down to grass, taking all the arable out of the system. Then we roll it out across the whole of England and evaluate pollution risk simulations for the whole of England using the Environment Agency funded National HYPE model. And you can see our 3 test catchments there nested within that. So we're right in the middle of this. And if you'd like to hear about this at a slower speed, then there is a publication out now which gives you far more detail on what is being done by the group up at Lancaster and with GABA Consulting. So the final thing I wanted to just say is we've also run a big stakeholder event in January of this year, and we used that to give a much more detailed presentation than I've been able to give you today. So if anyone's interested, I'm very happy to share those slide decks. There's 153 slides in that, so good luck with that. Have a big pot of coffee alongside you. But in the afternoon, we got together and we, with our stakeholders, and used their expert advice on what they thought would be most useful in terms of modelling products and which mitigation methods they thought might work best and which ones we should be testing in the model. So just want to acknowledge that that knowledge is going into that modelling effort. Thank you.